Imagine a genetic glitch, right? One that makes a 10-year-old child develop the hardened arteries, the joint stiffness, and the failing heart of an 80-year-old.
Yeah, it is a truly terrifying thought. But then, if you flip that completely around, imagine a microscopic worm in a laboratory back in 1993.
Right, the one that was genetically hacked to just, like, effectively cheat death.
Mhm.
It lived twice its normal lifespan without aging a day.
Exactly. We are looking at a biological landscape today that is incredibly messy, deeply fascinating, and ultimately it dictates how long and how well you are going to live.
Welcome to the Deep Dive. Our mission today is to explore the pathophysiology of aging and disease.
Oh.
We're taking a really hard look at exactly how the body changes over time and fundamentally why getting older is the ultimate vulnerability multiplier.
Yeah, and to get to the bottom of this, we are pulling from a massive stack of scientific history, evolutionary biology, and modern geroscience.
Because we aren't just talking about, cosmetic changes like wrinkles or gray hair.
No, not at all. We are looking at the actual molecular engine that drives disease, how we define aging itself, and the counterintuitive reality of why curing our biggest killer diseases wouldn't actually extend human lifespan as much as you might think.
Right. We're also looking at cells that act like zombies, a longevity drug they literally found in the dirt of Easter Island, and the massive failures of the supplement industry along the way. Okay, let's unpack this.
So the foundational step in this exploration is really reframing what getting older actually means for your physical body.
Right, because before we can look at how the cellular machinery breaks down, we sort of have to look at the macro level.
Exactly. In medical terms, aging is defined by a shrinking margin of safety.
And the source material gives us this brilliant concept for this. They call it homeostatic reserve.
Right, the homeostatic reserve.
So picture a bridge spanning a river. When that bridge is newly built, it has this massive weight capacity. It can handle rush hour traffic, heavy freight trucks, severe weather events.
Oh, sure. It absorbs all that stress and it just remains structurally perfectly sound.
Yeah, but as the decades pass, microscopic cracks form in the concrete.
The steel cables rust a bit.
Exactly. The bridge's absolute maximum weight limit starts quietly shrinking.
And the real danger there is that the bridge looks completely fine on a sunny day with light traffic. You would never know its reserve capacity is compromised until a major stress test happens.
And that exact same dynamic dictates human biology. Like, a 25-year-old kidney can filter blood at maximum capacity during a major medical crisis.
Right, like severe dehydration or physical trauma. And it can simply bounce back to normal a week later.
But an 80-year-old kidney, that's the older bridge. It might be functioning totally fine on a normal Tuesday, but it's already operating right near its absolute ceiling.
Yeah, so a minor stressor, a single bout of dehydration, a minor infection, or even just the contrast dye they inject for a routine CT scan that can exceed that shrinking weight limit.
And tip the kidney into total failure. The shrinking of the homeostatic reserve is what transforms a minor bump in the road into a fatal crash.
Which is wild because aging itself isn't technically classified as a disease in most medical systems, mainly because it lacks a single discrete cause.
Right. Yet it is the single strongest risk factor for the diseases that kill the vast majority of people in the developed world. Heart disease, cancer, stroke, Alzheimer's.
Yeah, and how to define the underlying mechanism behind that shrinking reserve? Well, that has been the subject of a fierce, century-long debate.
Right. The scientific community historically split into three distinct camps to explain this.
The first camp championed what they called programmed aging.
Which is the idea that aging follows a strict, genetically regulated schedule, kind of like puberty or embryonic development.
Exactly. And they point to the vast differences in the animal kingdom to back this up. I mean, a mouse has a maximum lifespan of roughly 4 years.
While a bowhead whale can live over 200 years.
Right. So if aging were purely random wear and tear, it would be really difficult to explain such wildly divergent, species-specific biological clocks.
But then the counter-argument was the damage accumulation camp, or stochastic aging, which pretty much dominated the 20th century.
Yeah, this camp viewed biology through the lens of thermodynamics. They basically argued, hey, there is no genetic program designed to kill us.
Aging is simply the inevitable buildup of unrepaired molecular damage over a lifetime. DNA mutations, misfolded proteins, cellular waste, it just accumulates until the system crashes.
But the third camp, they offer the most elegant, if somewhat brutal, explanation. In 1957, an evolutionary biologist named George Williams proposed a theory called antagonistic pleiotropy.
Okay, can you translate antagonistic pleiotropy into plain English for us?
I can try. So the core idea is that evolution is deeply pragmatic. Natural selection only really cares about your survival up to the point of successful reproduction.
Right. Once you pass your genes on to the next generation, natural selection is effectively blind to what happens to you.
Exactly. So Williams realized that a single gene can have multiple, completely contradictory effects at different stages of life.
So certain genes are incredibly beneficial when you're young and trying to survive a hostile environment to reach reproductive age.
Yes. But those exact same genes have wildly harmful side effects decades later when natural selection is no longer actively editing the genome, so to speak.
A classic example in the literature is the p53 gene.
Oh, P53 is a great example.
Right. In your youth, p53 acts like a superhero. It's this powerful tumor suppressor. Whenever a cell starts dividing erratically, p53 steps in and shuts it down.
Which prevents you from developing cancer early in life. This ensures you survive long enough to reproduce.
But decades later, that same aggressive tumor-suppressing action becomes a huge liability. It forces so many cells to stop dividing that it accelerates the exhaustion of your stem cells.
Yeah, it actively drives tissue aging and depletes your homeostatic reserve.
Wow.
What's fascinating here is how modern science has really moved beyond fighting over these three distinct camps.
Right, the field of geroscience has sort of synthesized these ideas. We no longer view aging as a passive, inevitable decline.
Exactly. It's now recognized as an active, measurable cluster of specific interconnected molecular mechanisms. And if they are specific mechanisms,
Then they can potentially be altered.
precisely.
To understand how scientists figured out they could alter the aging process, we have to look at the moment they discovered a literal ticking clock inside human cells.
We have to jump back to 1961 at the Wistar Institute in Philadelphia.
Right. An anatomist named Leonard Hayflick was culturing normal human fetal cells in a glass lab dish.
And the prevailing dogma of the time, established decades earlier by a Nobel laureate named Alexis Carrel, was that cells were inherently immortal.
Yeah, Carroll claimed he kept a culture of chicken heart cells alive and dividing for over 30 years. The whole scientific establishment believed that aging was a whole-body phenomenon.
We thought it was a failure of the macro system, not something built into the cells themselves.
And Carroll's chicken heart experiment cast a shadow over biology for half a century. Nobody dared question a Nobel laureate.
But Hayflick was noticing something strange in his own meticulous cell cultures. His fetal cells weren't dividing endlessly.
Right, Hayflick proved that normal human cells actually stop dividing after roughly 40 to 60 divisions. They just permanently halt.
Yeah, and it turns out Carroll's supposedly immortal chicken heart culture only survived for decades because his lab assistants were unknowingly adding fresh embryonic stem cells to the nutrient broth every single day.
Wow. So Hayflick exposed the flaw and completely shattered the dogma. His discovery, which is now known as the Hayflick limit, proved that aging is fundamentally a cellular phenomenon.
But wait, how does a cell know it's reached 60? It doesn't have a calculator. Well, the physical mechanism behind the Hayflick limit wasn't discovered until the late 1970s, and it's a breakthrough that eventually won the 2009 Nobel Prize.
The cellular clock is driven by structures called telomeres.
Yes, you can picture telomeres like the little plastic caps at the ends of your shoelaces, protecting the genetic material from fraying.
Right, the aglets of the chromosome.
Basically, yeah. Telomeres are repetitive sequences of DNA that cap the ends of your chromosomes.
And the biological flaw lies in the enzyme responsible for copying your DNA before a cell divides, which is called DNA polymerase.
Right. This enzyme cannot copy the chromosome all the way to the very tip. It requires a tiny runway to attach to.
Meaning with every single cell division, a small piece of the telomere is lost. The cap just gets shorter and shorter.
And once those telomeric caps get critically short, the cell recognizes that its actual structural DNA is in danger of being chewed up.
So to prevent catastrophic mutations in cancer, the cell pulls the emergency brake. It stops dividing and enters a state called cellular senescence. It's a brilliantly elegant mechanical timer.
It is, but around the same time, another theory of cellular aging emerged that ended up leading millions of people completely astray.
Oh, you mean in 1956, when chemist Denham Harman proposed the free radical theory of aging?
Yeah, the origin story is wild. Harman reportedly came up with the idea after reading a magazine article about free radicals in radiation chemistry while he was working at a Shell oil lab.
Right. So Harman proposed that aging is the result of cumulative damage from reactive oxygen species.
These are highly reactive molecules generated as normal byproducts of our own metabolism.
Our mitochondria, the cellular powerhouses that turn food and oxygen into energy, They are constantly leaking these free radicals.
And those free radicals bounce around the cell, damaging DNA and oxidizing proteins. Essentially, our mitochondria are slowly rusting us from the inside out.
The logic seemed incredibly intuitive. I mean, if free radicals cause aging, then swallowing high doses of antioxidants like vitamin C or vitamin E should neutralize them and extend lifespan.
And this theory birthed a massive multi-billion-dollar supplement industry.
But the clinical trials over the subsequent decades were a massive failure.
Yeah, pumping the human body full of antioxidant supplements did not extend lifespan. And in some trials, it actually increased mortality.
Wait, really? It increased mortality?
Yeah. The failure revealed this fascinating biological paradox. A low level of oxidative stress from free radicals is actually necessary.
Oh, because it acts as a fire drill for the cell.
Exactly. When a cell senses a few free radicals, it activates its own powerful internal defense systems. It makes the cell stronger and more resilient, a concept called hormesis.
So by indiscriminately flooding the body with external antioxidant pills, we were basically putting out the fire drill. We were silencing the crucial signals our cells need to maintain themselves.
Precisely. And the spectacular failure of the free radical theory forced a complete rethinking of how we categorize the biology of aging. Scientists realized they couldn't just hunt for a single villain.
Right. They needed a comprehensive map of the entire breakdown process. And in 2013, researchers published a landmark framework called the Hallmarks of Aging, which was just recently expanded in 2023.
Yeah, this framework has become the central organizing map for the entire field of geroscience. It identifies the specific molecular mechanisms that interact to drive aging.
Telomere attrition is a major hallmark. Genomic instability is another, mapping how the accumulation of DNA damage from UV light, chemical exposure, and replication errors just overwhelms the cell's repair mechanisms.
Massively raising cancer risk. Stem cell exhaustion is also on the map, explaining why the cellular reservoir responsible for regenerating tissue simply dries up.
Leaving older individuals unable to heal wounds or repair organs efficiently.
But the hallmark that has completely revolutionized how we approach therapeutic interventions is cellular senescence.
Right. When a cell reaches the Hayflick limit and its telomeres are depleted, it enters senescence. It stops dividing, but it doesn't undergo programmed cell death.
No, it doesn't.
This is where we get to the zombie cells.
Yeah.
Because the senescent cell is like, well, it's like that one highly toxic coworker who refuses to retire or quit.
That is a very accurate analogy.
They stop doing any productive work for the company, but they just linger around the office, loudly complaining, spreading negativity, and actively poisoning the morale of everyone else around them.
Right, and the scientific equivalent of that toxic complaining is known as the SASP, the senescence-associated secretory phenotype.
That's a mouthful.
It is. This term was coined by biogerontologist Judith Campisi. It describes how these zombie cells begin actively secreting a highly toxic cocktail of inflammatory molecules, cytokines, and tissue-degrading enzymes.
So they aren't just sitting there passively, they're actively chewing up neighboring healthy tissue and convincing nearby healthy cells to become senescent too.
If we connect this to the bigger picture, this localized cellular toxicity feeds into a massive systemic hallmark known as inflammaging, a concept introduced by Claudio Francesi in 2000.
Inflammaging. It describes how the aging immune system basically drifts into a state of chronic, low-grade, sterile inflammation.
Yeah, even when there is no viral or bacterial infection present, the immune system is constantly dialed up, reacting to the molecular garbage spewed by those senescent cells.
And this persistent simmering inflammation is now recognized as a primary driver of downstream catastrophes like atherosclerosis, where blood vessels harden, and Alzheimer's disease, where the brain's immune cells turn destructive.
Exactly. The realization that aging is active sabotage from within rather than passive decay changes everything.
Right. Once science mapped out these specific hallmarks, researchers began hunting for proof that manipulating a single hallmark could actually alter the speed of the aging clock.
And nature provides some devastating natural experiments that prove these hallmarks dictate health. The most profound example is Hutchinson-Gilford progeria syndrome.
It's an exceptionally rare genetic condition affecting roughly 1 in 4 million births.
Yeah, children born with progeria age at a terrifying 5 to 8 times the normal human rate.
By the time they are 10 years old, they exhibit the physiological markers of an octogenarian: severe hair loss, joint stiffness, loss of subcutaneous fat, profoundly hardened arteries.
And tragically, most of these children die from sudden strokes or massive heart attacks by age 13 or 14.
The mechanism behind progeria actually isolates a single hallmark, right? Genomic instability.
Yes, it is caused by a single letter mutation in the LMNA gene. This gene is supposed to produce a structural protein called lamin A, which acts as the scaffolding holding the cell's nucleus together.
But the mutation produces a toxic, warped version of the protein called progerin. And this warped scaffolding causes the nuclear envelope to literally collapse and buckle.
Exposing the DNA to massive, rapid damage. Progeria proves, in tragic fast-forward, that breaking a specific cellular mechanism directly drives the cardiovascular diseases that eventually kill most humans.
It is an unbroken line from a molecular defect to a fatal systemic disease.
Yes.
But here's where it gets really interesting. If a single mutation can catastrophically accelerate aging, could a single mutation dramatically slow it down?
Well, in 1993, a biologist named Cynthia Kenyon at UCSF provided the answer. She used a tiny transparent roundworm called C. elegans.
Kenyon altered a single gene in the worm called the DAF2 gene.
Right, and the DAF2 gene regulates a pathway that is remarkably similar to the insulin and IGF-1 signaling networks in humans. This pathway acts as a nutrient sensor.
So when food is abundant, the pathway signals the cells to grow and reproduce. But Kenyon discovered that mutating the DAF2 gene dialed down this signaling.
Effectively tricking the worm's biology into thinking it was living in a state of starvation and stress.
And in response to that perceived stress, the worm cells stopped prioritizing growth and started prioritizing maintenance.
They activated a whole suite of protective genes that repaired DNA, cleared out cellular garbage, and fought off oxidative stress.
So Kenyon's mutated worms didn't just live a few days longer. They completely doubled their lifespan.
And crucially, they weren't just lingering in a frail, decrepit state. The mutated worms remained energetic, youthful, and healthy long past the point when normal worms had died.
It was a massive extension of health span, not just lifespan.
Yes, and Kenyon's discovery proved that the rate of aging is malleable. It sparked the modern longevity boom.
But translating genetic miracles in microscopic worms to complex mammals is notoriously difficult. The messy reality of mammalian biology was perfectly captured in the great monkey diet drama.
Oh, the dueling monkey studies. This is such a classic example.
Right. Because manipulating the insulin pathway in worms, simulating starvation, researchers theorized that actual caloric restriction, eating about 30% fewer calories while maintaining optimal nutrition, would extend lifespan in primates.
So 2 massive, decades-long studies were launched using rhesus monkeys.
In 2009, the University of Wisconsin published their highly anticipated results. The monkeys on the restricted diet experienced significantly less cancer, lower rates of heart disease, and physically look years younger than the control group.
It seemed like a massive validation of caloric restriction.
But then the scientific community was stunned in 2012 when the National Institute on Aging published the results of their parallel monkey study.
The NIA study found 0 significant survival benefit.
None at all.
None. The calorie-restricted monkeys lived no longer than the normal diet monkeys. 2 teams of world-class scientists performed the exact same intervention on the exact same species and got completely opposite results.
When researchers finally performed a joint reanalysis in 2017, they discovered the discrepancy came down to the control groups.
Yeah, this is where it gets crazy. The Wisconsin study fed their control monkeys a purified lab diet that was extremely high in sucrose.
Basically feeding them junk food.
Exactly. So the restricted monkeys in Wisconsin weren't just eating less, they were eating less sugar.
Whereas the NIA study fed their control monkeys a naturally sourced, highly nutritious whole food diet. The baseline health of the NIA monkeys was already optimized.
So restricting their calories further offered no additional survival benefit.
It is a really humbling lesson for geroscience. Aging studies are just exquisitely sensitive to methodology, genetics, and baseline conditions.
Yeah, translating these interventions is fraught with complexity, which contributes heavily to the massive misconceptions the general public holds about our progress against aging.
Right. The biggest misconception is born from looking at historical charts. People see that average human life expectancy skyrocketed from roughly 47 years at the turn of the 20th century to nearly 80 years today.
So the assumption is that modern medicine is successfully slowing down the aging process. But we aren't.
The vast majority of that statistical gain came from eliminating early life mortality. The advent of clean water, modern sanitation, childhood vaccines, antibiotics, these stop people from dying of infectious diseases in their youth.
We drastically improve the odds of a human reaching old age.
But we have not pushed the absolute ceiling of the human biological clock. The maximum verified human lifespan remains 122 years. a record set by Jean Calment of France, who died in 1997.
Yeah, we are getting more people to the finish line, but we haven't actually moved the finish line itself.
Which leads directly into the actuarial concept of competing risks. And this shatters another major misconception.
Most people assume that if we could just cure cancer or completely eliminate heart disease, average human life expectancy would skyrocket.
But the math just doesn't support that. The underlying homeostatic reserve is still degrading. Like, if medical science miraculously cures your cancer at age 82, your underlying cellular aging hasn't been halted.
Your immune system is still suffering from inflammaging. Your telomeres are still critically short.
Your cellular vulnerability is still incredibly high. If cancer is removed from the equation, your degraded reserve guarantees that another disease, maybe a stroke, severe pneumonia, or Alzheimer's, is waiting in the wings and will take advantage of that frailty shortly after.
Right. Eradicating any single major disease would only modestly extend the average human lifespan because the underlying molecular engine of aging is still driving you toward failure.
And this actuarial reality is exactly why modern geroscientists are trying to completely rewrite the rules of modern medicine. They argue that playing whack-a-mole with individual diseases is mathematically inefficient.
Instead, they want the FDA to classify aging itself as a treatable indication.
Researchers like Dr. Nir Barzilai are leading this paradigm shift with initiatives like the TAME trial targeting aging with metformin.
Yeah, metformin is a remarkably cheap, globally prescribed drug for type 2 diabetes.
And observational data suggested that diabetics taking metformin were mysteriously outliving non-diabetics who weren't on the drug.
Right, so the TAME trial is designed to prove that administering metformin can delay the onset of multiple age-related morbidities simultaneously: heart disease, cancer, cognitive decline, by targeting the underlying cellular aging pathways rather than the specific diseases.
And the pharmaceutical landscape extends far beyond repurposed diabetes drugs. There is massive investment in senolytics,
Which are a class of drugs designed specifically to hunt down and induce apoptosis cell death in those toxic inflammatory zombie cells.
Then you have compounds like rapamycin. The story behind rapamycin is incredible. In 1968, a Canadian medical expedition took soil samples from Easter Island, known locally as Rapa Nui.
They discovered a bacterium in the dirt that produced a potent compound capable of inhibiting a fundamental nutrient-sensing pathway in cells, a protein complex now known as mTOR.
Originally, rapamycin was developed as a powerful immunosuppressant to prevent patients from rejecting transplanted organs.
But researchers later realized that by inhibiting the mTOR pathway, rapamycin effectively mimics the exact same cellular state of starvation and stress resistance that Cynthia Kenyon triggered in her worms.
And that caloric restriction triggers in mammals. Today, rapamycin is one of the most rigorously validated longevity interventions in animal models, repeatedly extending the lifespans of mice even when administered late in life. So what does this all mean for us today? Because as exciting as senolytics and mTOR inhibitors sound, it feels necessary to push back a bit on the hype. Biology rarely offers a free lunch.
No, it really doesn't.
Like if we unleash a senolytic drug into the human body to indiscriminately wipe out every single senescent zombie cell, aren't we taking a massive risk? Senescent cells aren't purely evil.
No, they're not.
Doesn't the body rely on some of those inflammatory signals to jumpstart the immune system during acute wound healing, or to permanently shut down a cell that is on the verge of turning cancerous?
This raises an important question that the field is actively wrestling with. The biological networks governing aging are defined by delicate evolutionary trade-offs.
Wiping out the SAESP with a pharmaceutical sledgehammer might alleviate chronic inflammaging, but it could simultaneously cripple the body's ability to heal a surgical incision or contain a microtumor.
Yeah, intervening in a system as complex as human homeostatic reserve requires surgical precision. We are still in the difficult phase of translating these spectacular mouse miracles into safe, highly calibrated human therapies.
We have covered incredible territory today, from Hayflick's shattered dogmas and the chemistry of free radicals to the monkey diets and the Easter Island soil. All of this laboratory science ultimately converges on massive real-world implications for everyone listening.
Because the demographic reality is staggering. We are currently living through the most dramatic demographic shift in human history.
By the year 2050, roughly 1 in 6 people globally will be over the age of 65.
And if we do not figure out how to intervene in the pathophysiology of aging, the sheer scale of chronic disease, dementia care, and the collapse of homeostatic reserve will place an insurmountable burden on global healthcare systems and labor markets.
On a deeply personal level, the goal of all this science fundamentally aligns with a concept introduced in 1980 by a Stanford researcher named James Fries. It's known as the compression of morbidity.
Right. The core philosophy behind geroscience and the goal of evidence-based lifestyle choices like maintaining muscle mass through resistance training and optimizing sleep is not simply to drag out the dying process.
We are not trying to add an extra decade of frail, bedridden existence to the end of your life.
Exactly. The objective is to square the curve. You want to maximize your health span.
You want to maintain the high cognitive function, physical independence, and deep homeostatic reserve of a much younger person for as many decades as possible, followed by a very rapid, compressed period of decline right at the very end.
The ultimate goal is to shrink the morbidity window, keeping the biological machine running at optimal capacity until the very end, avoiding that prolonged, tragic decade of chronic deterioration.
Understanding that dynamic changes how you view everything from daily habits to the arc of your entire life. So, we want to leave you with a final thought to mull over today.
Yeah.
If modern geroscience succeeds in fully compressing morbidity, meaning you could fundamentally maintain the biological vitality, the limitless energy, and the resilient homeostatic reserve of a 30-year-old right up until the closing months of a 100-year life, how would that reality fundamentally change the way you plan your career, your retirement savings, or even the timeline of your long-term relationships? Until next time, keep exploring.